and is the most recent common ancestor of land plants. The problem is, it looks so ignominious. It's just this pathetic little thing, and all it is this structure going on top of this big, huge thing. This is a water lily leaf. So you have an aquatic plant, and these guys grow as epiphytes on aquatic plants. So they grow fully submerged in the water, growing on, sometimes on rocks, but usually on submerged aquatic vegetation. Epiphyte, everyone good with that? Plant that grows on another plant without parasitizing it? Okay. So all you see is a little disc of shells. Since this is a section, you just see, a section of cells, single cell layer thick, and then a number of cells that are arranged in a disc-shaped pattern. That's coleochaete. Okay, what's the big deal? The big deal is, this was my algae professor in Madison. She got famous for discovering this. She got an electron microscope and looked at the cell division in coleochaete, and she found that they have a phragmoplast and they form plasmodesmata. That is astounding. Do you remember those words? Phragmoplast is an extra set of microtubules formed late in mitosis, and it helps direct vesicle towards the forming cell plate. And then as cell division ensues, the little holes that are left in the cell plate by those remnant microtubules from the phragmoplast become holes between the mature cells. Those are plasmodesmata. They allow for movement of substances between cells. So you do not see that in the other algae, most other algae. And of course they don't need it. Why? Because they are aquatic algae and they exchange nutrients, water, carbon dioxide, all the nutrients, everything they need directly with their environment. Okay, so plasmodesmata allows for formation of parenchyma. Parenchyma was otherwise thought to be unique to land plants. We discovered plasmodesmata forming parenchyma in coleochaete and got very excited. Okay, so the question is, I know you share my excitement back there, Christine. Yeah, okay, so the question is, what is it doing? The adaptive significance shifts. So when you have something that evolves for one purpose, and then its function changes, we call that a pre-adaptation. So the pre-adaptation for coleochaete is allowing nutrients to move from one cell to another so that nutrients from these cells can move into the egg cell and then later the zygote as it undergoes sexual reproduction. All right, so this guy is fine just taking water in and out from each cell by cell and carbon dioxide, oxygen, all that kind of stuff. But the adaptive significance of parenchyma and coleochaete is really just so that the nutrients can move as this thing is dying. Remember, this water lily leaf is going to die at the end of the growing season. And then this guy's saying, okay, I'm going to make a zygote that'll persist over the dormant season. So the real analogy is they get stressed and they have sex. Right? So when things are good, they're not stressed, they're just growing, they have mitotic divisions. At the end of the growing season, like, oh no, this is getting harsh. And then they'll make an oogonium that makes an egg, and then another plant will make sperm, you'll have fertilization, a zygote, and then all the nutrients move into the zygote through the plasmodesmatal connections. Okay. In this one, you can just see the whole plant. So it's a whole mound. It's just a single layer of cells, just less than a millimeter across. Also, what's interesting here is that these cells divide here, and the thing gets bigger by divisions here, and it kind of spreads out. That's like an apical meristem. Right? So cell division in algae doesn't occur through apical meristems either. So now we have the evolution of an apical meristem in coleochaete as well. All right, let's move on. I'll just do these Anthopteris. First of all, this is upside down. There is a rounded foot, and then you can see all this dark stuff. That is extra surface area in the cell wall. So there's all these different interstices in the cell wall, and that allows for extra surface area for conduction of nutrients from the gametophyte into the foot, and that transfers to the sporophyte. This thing's upside down. So here's the thallus. This thing here is called an involucre. It's just a little collar that forms around the base of the sporophyte. So here's the foot. There really is no seta. Instead, there's an intercalary meristem. Okay, meristem, region of dividing cells, intercalary, placed in between the foot and the sporophyte. What this means is you had an archegonium here that made an egg that got fertilized, it turned into a zygote, the zygote divided, then formed a sporophyte. And then what that means is if you have cells growing here, as they push upwards, they differentiate, you get spores that form by meiosis. You can get indefinite spore production from a single fertilization event. Fertilization in bryophytes is limiting because it requires an external film of water in an aqueous environment. Okay? So this is how hornworts survive, even though they're otherwise quite simple and, dare I say, pathetic plants. No, I didn't say that. You're going to edit that out. Okay, so now what else can we do here? Here's the spores. You can see there's groups of 4 spores together. Those are called tetrads. And then what's hard to see, I'm not sure if I have a good side, there's also eliders that are formed with the spores. So the spores are formed by meiosis. There's also specialized cells that are formed next to the spores, and they are cylindrical cells that have helical wall thickenings. So there's cellulose that's in basically a spiral. And then as this thing splits open, as it pushes up, it gets older, and the tissue just kind of deteriorates and dries out, and then the spores are exposed. As the eliders are exposed to drying currents of air, changes in humidity, that's called hygroscopic. Hygroscopic is like sugar getting hard in your kitchen in the summertime. It absorbs water. It also loses water, right? So cellulose can absorb and lose water. Changes of movement causes those eliders to twist and then pushes those spores out and facilitates dispersal. My favorite way to remember it is the packing peanuts principle of spore dispersal. Is that going to work for you? Right? Packing peanuts provide buoyancy to your shipment from FedEx or whatever, right? So this provides buoyancy. Like eliders, what does elated mean? You guys were elated to come into your botany class today, right? Yeah. All right, so see what you can do with that. We'll see some more eliders in a bit. All right, that's just the tip. It's the whole mountain. You can see the spore. This is a young sporophyte. It hasn't even started splitting open yet. So that's the very tip of the hornwort. Okay. Thallus. Thallus is just pathetic. It's just parenchyma cells. There is no cuticle. There's no differentiation in the thallus either. So not much to talk about there. If we had one that was from the water, like the ones in back there, you would find blue-green algal cells growing in between the cells of the thallus. So that's a mutualistic symbiosis. What is exchanged? All right, compare it to mycorrhizae. So fungi living on the roots of trees, that's mutualistic symbiosis. The tree would give sugars to the fungus would give mineral nutrients to the plant. Algae and hornworts, they're both photosynthesizing. It's not like the hornwort needs sugar from photosynthesis that the algae is providing. What is the blue-green algae doing? What do? Microbes can only do this. Fix nitrogen. They have nitrogen fixed, so they are providing nitrogen for this. Okay. Polio. Marcantia. All right, you got to tell the boys from the girls. Marcantia is the genus you'll learn for the complex thalloid liverworts. And then we're looking at the apex of an anthoridiophore. So you learn the words antheridia and archegonia for all of these plants, okay? But you learn antheridiaphore and archegoniophore for Marcantia in particular. What does it mean? It's a structure that bears the antheridia. In this case, it looks like an umbrella. So there's a stalk that arises from the thallus, and then pointing upwards, we have antheridia. Those are round here. And then they're pointing upwards. What that means is that eventually they get bigger and these cells start breaking down. And then the force of a falling raindrop is a huge deal in the life of one of these liverworts. All right, that's going to have an impact. It's going to rupture the antheridia at precisely the time when, hey, it's raining, meaning there is available water to facilitate dispersal. So I want you to look at your Raven diagram here. He's got individual raindrops splitting off, splashing off of the antheridiopore, carrying the little baby sperm with them. Note the attention for detail in Peter Raymond. I mean, he didn't have to do that. And you look at those sperm, they even have racing strips on them. Those guys are built for speed. I'm telling you. Okay. I just want you to appreciate the finer things in life, like those 2-page diagrams in Peter Raymond. That is just amazing. Okay, so here we go. This one is a mess because you're just making a slice of it. This is the antheridiopore. What you have to learn is how to find an archegonium. That's here. And what you're looking for, there's going to be variations as you go from one group to another, but you're always going to have a fat egg cell, and then surrounding it are venter cells, right? And then there's a neck. So a neck and a venter, it looks like a flask. Okay, so that's your basic thing. Find the archegonium. Note that they're hanging down. And then note there's rhizoids. These are absorptive cells that conduct water, and they're going to draw water all the way up this thing, because this sperm has to swim over from another Mercantia and then swim up the archegoniophore and then find its way into the archegonium and then fertilize. Okay? The reason these are elevated is to facilitate spore dispersal later. So it's all about water in these bryophytes, and there's ways that they need water for fertilization. When they form spores, they need to be dry and exposed to air. So they push up above that thing, the spores are released, the eliders work, it's drying out, and then the spores disperse. So it's this trick of how does it stay wet when it needs to, how does it dry out when it needs to. Usually something elongates, and that facilitates spore dispersal after fertilization. All right, so that was fun. Let's see if this Archegonia 2 is any better. Not so much. No. You see a little more of the seed, I guess. No, Gemmy. All right, Gemmy are asexual reproduction. So there's 3 things you learn on the thallus of Mercantia. The 2 sexual things, antheridium and archegoniophore, and then the Gemmy cup are asexual, meaning these guys are haploid, and they're just little discs of haploid tissue. They're formed inside the cup. And again, when it's raining, the force of a raindrop will cause this force to be concentrated at the focus of a parabola. You know how to learn how to determine that? Pre-calc? Oh, blank stares. Oh, you have satellite dishes at home? No, the cool kids have Hulu or something. All right, so you got a dish, and then you have something that sticks in it. That's the receiver. That's at the focus of the parabola. So there's a point right in here where all that energy is concentrated because it's reflected from this cup-like structure. That force of the falling raindrops ejects these gemmy. They go splashing off. The rain washes them away, and they turn into a new thallus by asexual reproduction. Sporophyte. All right, so you have to imagine this is hanging down from an archegoniophore. Here is the foot, it's rounded. The seta is a stalk. It doesn't elongate appreciably. Why? Because the archegoniophore is elongating. Here's the capsule that has spores and eliders inside of it. And then you have to think about changes in stages of development. Here is a calyptra. And you have to remember that the calyptra develops from the neck and the venter of the archegonium. So this thing has gotten much bigger, and that sterile jacket of cells surrounding the egg, after the zygote forms, the sporophyte forms, it gets stretched and stretched and stretched, and ultimately ruptures. Right? And then outside of that is what we call a perianth. Those are other leaves that surround that. So I can ask you, I can point to you that and say, what's the ploidy level? It's haploid. And then what's the term calyptra? What does it develop from? The neck and the venter of the archegonium. We'll see other examples of that. Thallus. All right, this one's upside down too. There's scales and rhizoids. The scales are multicellular. The rhizoids are unicellular. In most cases, they're coming at you through the plane of the section, so you just see a little circle that's cut in cross-section. Rhizoids are single cell, scales are multicellular. Here are parenchyma cells mostly storing water and not photosynthesizing. On the top surface, there are photosynthetic cells. On top of that are cells that have a heavy cuticle, and then there are pores. that allow for gas exchange. Make sure you see the conocephalum in back because the pores are really obvious.